Our next paper is titled, "Is Spinal Cord Ischemia Following Total Arch Replacement and Endovascular Stenting of the Descending Still an Issue? Pathophysiologic Investigations and a Porcine Model."
Hello, ladies and gentlemen, dear Chairman. The frozen elephant trunk technique is a single-stage hybrid procedure, as everybody knows, which combines two different procedures: an open surgical aortic replacement of the aortic arch with, of course, the neuroprotective techniques like hypothermic circulatory arrest and selective cerebral perfusion, in our days performed at not that deep hyperthermia, so at 28 degrees, mild hyperthermia, and an endovascular stenting of the descending aorta without any neurological protection. More than that, we have here a thrombotic occlusion of the thoracic segmental arteries, which is wanted. So here we produce a trauma. So, of course, we have also a higher risk of spinal cord ischemia caused to that. The paraplegia and paraparesis rate is between 1 and 16% in the literature. We heard today something similar, 5%. So it depends, of course, also of the experience of the surgeon and of the center. The objective of our study was to look at the evidence of spinal cord ischemia in a porcine model of frozen elephant trunk procedure with prolonged hypothermic circulatory arrest, which can occur, so 90 minutes we performed, with selective cerebral perfusion at 28 degrees Celsius, and the supradiaphragmatic occlusion of the thoracic segmental arteries. For this reason, we operated on 14 pigs, female pigs, which we divided into groups. All animals were monitored hemodynamically through the entire operation. We performed an incision in the intercostal space, connected the heart-lung machine, cooled the pigs down to the desired temperature of 28 degrees Celsius. At this temperature, we had 90 minutes of selective cerebral perfusion, so lower body ischemia. And in one group, and in the other group, we did the same, but also through another incision, occluded the TSA between T4 and T13, so between 8 and 9 thoracic segmental arteries in the pig. It depends, a small variability. And then we looked about the regional spinal cord blood flow, the motor evoked potential changes, and the extent of the spinal cord neuronal necrosis after reworming and going off bypass. Here are the results. The thoracic cerebral blood flow, the regional thoracic cerebral blood flow during selective cerebral perfusion, so lower body ischemia, was in the thoracic area. It was stable in both groups without differences. In the lumbar area, it decreased to 13 and 17, between 13 and 70% without differences between groups. But during the reperfusion, we see in both groups a hyperperfusion of the former ischemic area with a lower increase in the group where the TSA were occluded. And this difference was significant. The same we saw in the measurement of the amplitude of the motor evoked potentials. We looked at 4 different muscle groups, and the farther we went from the stimulus locus, so from the cerebral, the more distal we went, the more the effect was more prominent. The clipped group didn't recover. You can see it very well on every slide. Here we have a recovery, here we have a recovery, here we have a recovery. In the non-clipped group, where the clipped group has no recovery. And finally, we looked at the necrosis, and the extent of the necrosis was quantified using a Kleiman's schematic grading system published by Etz and collaborators some years ago. And we saw here a significant difference only in the lumbar part. So in the lumbar part of the spinal cord, we had much more necrosis, an extended necrosis which the difference was significant. In the thoracic part of the spinal cord, we have no difference. And now we make the conclusion that 90 minutes of selective cerebral perfusion provide enough spinal cord protection during the open arch replacement, even at 28 degrees Celsius. But in combination with the occlusion of the TSA, which is the case in a frozen elephant trunk procedure, the lumbar selective blood flow, the lumbar spinal cord blood flow, may be altered, which may cause decrease in MAPs and a higher necrosis rate in the neurons. So further investigations are required in order to indicate the critical extension of TEVAR. So how far do we go with our stents? And to answer the question if there is a use, in this case, for lower body perfusion during SEP. Thank you.
Yeah, thank you, Peter. Very sophisticated animal study again. There's two comments I have. One is the Hanover group published a couple of years ago their clinical results, and they found that the paraplegia mortality rate is 6 times higher once you go over 9